In genomics, we sequence the genome, the DNA complement of the cell. So that can be a whole genome sequencing project, WGS, or we can also do some specific areas where we call it as a partial genome sequencing project. So different types of genome sequencing can be, there can be de novo sequencing, there can be resequencing, or there can be targeted resequencing. So in de novo sequencing, we sequence a genome that has not been already sequenced. So we sequence it this first time, and then once we sequence it, since we do not have any reference, so we put it together, and that project involves what we call it as an assembly project. So we take those short sequencing reads and we align them on the basis of overlaps between those reads. So that thing is called as assembly. In resequencing, what we do here is we sequence some regions, we sequence the genome, and then we try to compare it with some reference. So in this way, we can correct the assemblies which are already there, or we can compare and identify new areas which may not be present in the old assemblies. In this way, we can also get the idea about the variations among these two. We can look into different mutations, etc. Like for example, we can have CNVs. We can idea about where we can have the idea about the copy number variations. We can also look for indels, which are insertions and deletions. So if some sequence is present in one assembly, we can say there is an insertion. So wherever it is missing, over there we can say there is an deletion. So both words put together we call them as indels. Whereas SNPs are single nucleotide polymorphisms. So sometimes we observe that just one nucleotide is different between these two, but that nucleotide may be crucial or critical. Maybe it's there in some codon of some important amino acid that is a part of the protein. So in this way, these SNPs may be important controlling different biological functions. So we can also look into the rearrangements, that is the arrangement of the genes on those chromosomes. So we can have segments rearranged in these two assemblies if we compare them. In targeted resequencing, we take just a subset of that genome. So in this way, we can be targeted, we can be specific, we can save our resources, and within less period of time, we can identify what factors we are looking for, and we can be kind of very specific in our approach. So we can target the exons, which are the protein, which are the coding regions of the GNA. So they give messenger RNA, and messenger RNA then gives the proteins. We can target whole chromosomes, or maybe we can target some set of genes. In targeted sequencing, the question is how to capture those target regions. So there are three different approaches. There are special baited probes which are designed for this. We can also do some array capture techniques which are already there. So we can use some arrays where we can have glass slides. On that we have the probes. So with the help of those probes, we can capture our target genes. We can also use PCR based methods. So we can amplify some specific regions and then we can use them to sequence. So here we see baited exome capture. So we start from here. So we have genomic DNA, and we have generated some random DNA sequencing reads where exons are shown in this orange color. We design some special capturing probes which are biotinylated. Bioteins are attached to them, and then these probes, they hybridize to the targeted exons. So they are complementary, and then they hybridize with one another. So in this way, we can capture where we have exons in which we are interested, so we can get these exons. And then they are applied to react with special beads, and then they attach over there. And in this way, we can identify them, we can take them out, and we can amplify them, and then we can send them for our sequencing. So we have captured some specific regions in which we were interested in this technique. We can also design some special microarrays, and with the help of those microarrays, we can capture our genes of interest. We can capture DNA, and then that DNA can be extracted. It is good for recovery of all targeted genes, but the drawback is that we need a large amount of sample. In PCR-based methods, we design specific primers against the genes of interest, those genes which we want to identify them, and then we amplify those regions. So once we amplify those regions, we can get those fragments out, and then we can submit them for sequencing. So how we can do mutation detections? Mutations are actually the changes in the genome that can be of different types, like there can be some copy number variation, so we can have more copies of some fragments or some genes. We can also have SNPs, we can also have indels. So how we can apply this next generation sequencing towards mutation detection? So if we look here in this slide, so here we have a reference genome, and we do have those all short reads, so we simply align them. So those alleles, they get those reads, they get aligned to that reference. And in the green, we can see there are some areas where we can have some mutations, where we can have maybe some single nucleotide polymorphism SNPs. So we compare what we have in the reference. So in the reference, for example, we have T's, but within those reads, we see there are some A's which are replaced. So in this way, while also taking into account the statistical tests, we can say that there is a SNP existing in this region. So in this way, we can also have the idea, if we take these reads and we align them, and in some region we see that the reads are not aligning, so maybe in our reference we have insertion, but the genome which we are comparing might have deletions in it. So what we conclude from here is that de novo sequencing is where we sequence a genome maybe for the first time, which is not already existing, whereas in resequencing projects, the assembly is already there, but we are still sequencing we are submitting new sequencing projects, and we are getting the sequences, and we are comparing them with the already existing assemblies to find those variations.